Today we are going to discuss the measures of variability or measures of dispersion. And this is also one of the fundamental concepts in descriptive statistics. So what is variability and why it is important in statistics? So variability stems from variation, which means being different. So it is basically about some trait that we are studying in a population, and we find that that trait is variable. That trait is different among different members of the population. So this is variability. This is the basic concept in statistics. If there were no variability within population, there would be no need of statistics, right? So what does it mean is that in statistics, we basically discuss about or basically analyze the presence or the behavior of different traits in the population, right? So why do we need to study those traits? Because those traits are expressed differently among different members of the population, among different units of the population. So variability is the very basic thing which is the foundation of statistics. So if there was no variability in traits, if the traits they were not variable, if the traits they were constant among the population, there was no need of statistics. And if the traits were not variable, if the traits were constant, then a single item or sample unit or the sampling unit that would give us all the required information about the population. So we would not need statistics if there was no variability, right? Because why do we study statistics? Because we want to study the behavior of the trait. We want to study the trends in the trait. So that is the reason we study statistics. That is the reason we use the statistical principles. But if something is constant, then there is no need for the statistical analysis if something is constant. For example, the number of eyes a human has, that is almost constant unless there is an abnormality, right? So you don't study the number of eyes as a variable in human population because that is something which is constant. Similarly, you don't study the number of fingers in human population usually because that is usually also constant, right? So the number of eyes or the number of fingers, so these are examples of those traits which are usually constant in the population. So they vary only in special circumstances, right? So in the normal circumstances, they are same. But if we talk about the skin tone, it means that this is a constant thing or is it a variable thing? So this is the trait in which we are talking about variability, because skin tone is not constant. Skin tone is variable, right? Although this is a qualitative variable and we don't actually deal with the qualitative variables as such in statistics, but this is just to show an example that the variability is the very basic foundation of using statistics. Like the height, because height is defined by the genetic makeup, right? So height is a quantitative trait. So height is variable. Not all human beings, they have all the same height, right? The number of eyes is constant in all of the human beings except in certain special circumstances, but the height is different, right? So every individual has got a different height. So this is something which is variable. And that is something that we can study in statistics, because if this is if this was constant, like all the human beings, they were all 5 feet tall, then there was no need to study that trait, because if we get information from only a single item, that information is sufficient, right? But why do we need to study the trait in different members of the population? Because that trait is variable among different members of the population, and that is why we collect information about that trait, and then we study that trait according to the statistical principles, right? So variability, the presence of variability is the very basis of the science of statistics. So variability is the thing that is the basic concept in statistics, and why variability is important. So it is not enough simply to give the measures of central tendency. The information about variability is also needed. Like in the previous lecture, we studied the measures of central tendency, that if a trait is variable and we are collecting information about that trait from the population, and then we want to identify, we want to find out a number from those observations that can represent all of those observations in the population. And that number is known as the measure of location or the measure of central tendency. So that is the number that is going to represent that trait in the entire population. But the information of that central number, information of that central tendency is not enough. We also have to identify that how much this central tendency varies. How much is the variation from this central number? Like if we are going to measure height of students and we identify their average height as 5.5 feet, right? So if their height is measured, the average height is measured as 5.5 feet. So is this enough information? No, we also are interested in knowing that what is the actual spread of this average height, how much this how much the units in statistical population, they differ from this central number, how much they vary from this central number. So this information about variability is also important, right? So not just we have to identify the central point, we also have to identify the variation from that central point, that how much it is variable. Is it variable to a larger extent or is it variable to a smaller extent? So this information is also necessary. And there are mathematically defined measures which are used for the quantitative analysis of variability. And the most basic of these mathematical measures are range, standard deviation, and variance. So these are the three most basic measures of variability. Now, starting from the range, right, which is the simplest measure of variability. So what is range? Range is simply the highest and the lowest observation. That what is the highest observation and what is the lowest observation. So range is simply the spread of observations, that what is the range of observations, starting from the lowest point to the highest point, lowest observation to the highest observation. So this is the simplest measure of variability, and this is range. So range shows how wide the distribution is over which the measurements are spread. Like we discussed in the previous slide, that in the measures of central tendency, we identify the central location. We identify the point where cluster of observations is located, right? What is the central point? What is the average point in our observations in our data? So what is range? Range actually gives us the entire spread of distribution, that from where to where our observations are located, right? So in central tendency, we are identifying the central number, but in the range, we are also identifying the spread of observation, the distribution of observations, that the central number, for example, if the central number is 6 in some case, right? So if we are giving the range that the central number is 6, but the range is from 1 to 11, let's suppose, right? So range gives us the spread of observation, that how widely the observations are spread, right? So this is the simplest measure of variability, and this is known as range. Now let's see just some simple examples of range. For example, we have the length of 100 shoots with us. And what we have to do is we have to define the range, and for that purpose we need to identify the lowest observation and the highest observation. The lowest observation is 68 and the highest observation is 80. So what we are going to write down in the result statement is that the shoot length ranges from 68 to 80 millimeters, right? So this is how simply we can define the range, that the range of this set of observations is from 68 to 80. So the shoot length ranges from 68 to 80 millimeters, or we can also say that the range of shoot length is from 68 to 80 millimeters in our sample. Then we have the other example, which is the masses of 50 salmons, and there also we have to identify the lowest observation and the highest observation. And this can be written as salmon mass ranges from 156 to 200 grams, right? So range is simply, there is no calculation needed, you just have to identify two numbers, the lowest observation and the highest observation, and that's it. So this is the simplest measure of variability. However, range, although it describes us or it gives us an idea that how wide the distribution is, but it doesn't give us an idea about the pattern of distribution. That what is the pattern of distribution? Are the observations evenly spread, or are they clustered around one point, or are they clustered around 2 points or 3 points or more? So what is their distribution, right? So range only gives us about gives us the idea about the width or about the spread of observations, that how wide the observations are distributed, but it doesn't give us an idea that what is the pattern of distribution within that range, right? So this is range. And here is the most widely used measure of variability, right? This is known as standard deviation. So this is the most widely applied measure of variability, and it is calculated directly from all observations of a particular variable. Like in the range, we were only picking up two numbers, the lowest observation and the highest observation, that's it. But standard deviation is actually a calculation that how much each observation deviates from where, how much this observation deviates from the central point, right? So this is that is why it is known as standard deviation, and that is why it is calculated directly from all observations of a particular variable. And this is the formula for standard deviation calculation from population, and this is the formula for the calculation of standard deviation from the sample, right? So here you can see that we have different symbols. So in case of population, the standard deviation is defined by the symbol sigma, and sigma refers to the population standard deviation, mu refers to population mean, x is a single observation, and n is the total number of observations. And x minus mu is deviation of an individual value from the mean value. And similarly, in case of the sample standard deviation, we can see that small s represents the sample standard deviation, x bar represents the sample mean, and x represents a single observation, n represents the total number of observations in our sample, and x minus x bar is deviation of an individual observation from the mean value, right? So this is the these are the formulae for calculation of standard deviation. And one more thing that you can see that the formula for calculation of population standard deviation is a little different from the formula for calculation of sample standard deviation. In the population standard deviation, we divide the deviation from mean value by the total number of observations. But in case of sample, we divide the deviation from mean value by n minus 1. So this is a slight difference in their formula. Otherwise, rest of the calculation in standard deviation is same for population as well as for sample. Now we discuss the term x minus x bar. So as we discussed in the previous slide, that x minus x bar represents deviation from mean, deviation of individual observations from mean value. So this term is known as the x minus x bar. This term is known as deviation from mean. When we take the square of this deviation, this is known as square of deviation, right? Square of deviation from mean, or simply the square of deviation. And when we take the sum of square of deviation of all individual observations in the sample, so their sum is known as the sum of squares of deviation, or simply the sum of squares. So this is a very popular term in statistics, and it is used in many statistical tests, and not many, in a number of statistical analyses. And this term is known as the sum of squares, right? So what is sum of squares? You calculate the deviation of individual observations from the mean value, then you take the squares of those individual deviations from the mean value, and then you take the sum of all those individual observations deviation from the mean value, and that sum is known as the sum of squares, right? Now we discuss the calculation. Now we are going to see that how this deviation from the mean value is calculated. And for that purpose, we are going to take an example about the shoot length. So we have 10 observations of shoot length, and these are the 10 observations that you can see here. And what is the formula? And we are supposing that these are the samples, so we are going to use the sample standard deviation formula for this purpose, and this is the formula. And in the formula you can see that first we have to calculate the mean value, right? Because the standard deviation is deviation from mean value, so it means that calculation of mean value is the first thing to do when you are going to calculate the standard deviation, right? Now we are going to see that how we can do this calculation. So we are going to make a table for the calculations, and in the table we are going to arrange these observations in an ascending order here. So in the first column we have just the number of observations, and in the second column we have their x values, the individual observations. Then in the next column we have x minus x bar, that how much each individual observation varies from the mean value. And then we have the next column x minus x bar whole square, which is the square of deviation from mean value for all the individual observations, right? So as we discussed in the first slide of standard deviation, that standard deviation is a statistic that is calculated directly from each of the observations. So here we can see that we are going to calculate the deviation of all the individual observations, right? So first of all, we have to calculate the mean value because we are going to calculate deviation from mean, right? So we have to calculate the mean value first. And this is the formula for the calculation of mean value. And for that purpose, we need summation x. So we are going to calculate the sum of all observations, and this is 814. Right, so summation x in this case is 814. And what is the number of n? What is the total number of observations? We have 10 observations. So we are going to put in the values to calculate the mean value, and it turns out to be 81.4, right? So 814 divided by 10 is equal to 81.4. So this is the mean value. In the next step, we have to calculate deviation from mean value for all the individual observations. Why? Because that is the requirement of the standard deviation formula. So here we have this calculation. So 78 is our first observation and 81.4 is our mean value. So our first calculation is 78 minus 81.4 in this column. Then our next calculation is 79 minus 81.4, then 80 minus 81.4, and so on. So we calculate the deviations of all the individual observations from the mean value, that how much these individual observations they deviate from the central point, how much they deviate from the mean value. So 78 minus 81.4, 79 minus 81.4, and so on, and we get the results. So 78 minus 81.4 is minus 3.4, 79 minus 81.4 is minus 2.4, 80 minus 81.4 is minus 1.4, and so on. So we calculate these deviations for all the individual observations. So you have to ignore this negative value here, because in the next column we are going to take the square of these individual deviations from mean values. So we are going to take the square of minus 3.4, so this is 11.56. Then we have the square of minus 2.4, and this is 5.76. Then we have the square of 1.4, this is 1.96. Again 1.4, 1.96. Then we have the square of 0.4, which is equal to 0.16. So we are going to calculate the square of all the individual deviations from the mean value, right? So now we have calculated the square of deviation from mean value for every single observation in the sample. So what is the next? We have to take their sum. So we are going to take the sum of this column, and that is going to be our sum of squares, right? So this term is the sum of squares, which is the sum of this column. So we have taken the sum of all these 10 square of deviation from the mean value, and we have our sum of squares as 56.4. Right? So now we have all the information which is required for the calculation of standard deviation, and now we are going to calculate this value. So here we are going to put in the values of 56.4, which is sum of squares divided by 10 minus 1, and the standard deviation is 2.5. Now we have to report our results in a proper statement form, and for that purpose we are going to write down the results as Mean plus minus standard deviation chute length is 81.4 plus minus 2.5 millimeters. Now remember that the term standard deviation is deviation from something. So deviation from what? Deviation from the mean value. So therefore, whenever you're going to report the result for the standard deviation, you have to report what is the mean value. So this is going to be useless, or this is going to be incomplete statement if you're not including the information from the mean value, that what deviation you're talking about, right? So you're talking about deviation from something. So what is this something? So this something is the mean value. So therefore, you have to write down the mean value along with the calculated standard deviation. So this is one form in which you can present your results, that mean plus minus. So why plus minus? Because you can see that some values, they are in negative, right? So they are lower than the mean value, and some values, they are higher than the mean value, the plus sign. So that is why we are mentioning both of these signs. So mean plus minus standard deviation chute length is 81.4 plus minus 2.5 millimeters. So this is the calculation of standard deviation. Now we calculate the standard deviation for grouped data. In the previous example, we calculated standard deviation for the ungrouped data, where the number of observations were few, so we calculated directly from the ungrouped observations. But what if we have a large number of observations and these observations are present in the form of a frequency table? So how we are going to do that? So we have another formula for standard deviation in that case, and this is summation f x minus x bar whole square divided by summation f minus 1. So this is basically the same formula, that we have to take the square of deviation of all the individual observations, and then we have to divide them by the number of observations, right? So we are going to take this example in which we have 100 observations, and we have information about their x values, and we have information about their frequencies, right? So this is a grouped data, and we are going to calculate standard deviation for this one. So here we have our standard deviation calculation table, and in this table you can see the first column is for the x value, the next column is for the f or the frequency values. Then in the next column we have fx, which is the summation fx, which is required for the calculation of mean value, and then we have the columns for the calculation of deviation from mean value. So here we have the formula for mean value for the group data, because we need to calculate the mean value first. So for that purpose, we need the summation fx value and we need the summation f value. So summation f is 100 and the summation fx is 1 multiplied by 68, because fx means frequency multiplied by x. So 1 multiplied by 68, 2 multiplied by 69, 4, which is the frequency of class number 3, which is represented by the observation 70. So 4 multiplied by 70, then 7 multiplied by 71, and so on. So we are going to calculate the individual fx values, and here we have these calculations: 68, 138, 280, 497, and 792, which is 11 multiplied by 72, and so on. So we have these calculations for all these individual values, and then we are going to take the sum of this column, which is going to be our summation fx. And summation fx in this case is 7400. Now we have both of the values, so we are going to calculate the mean value. 7400 divided by 100 is equal to 74. So this is our mean value. Now we are going to use this mean value for the calculation of deviation from the mean value. And starting from the first column, we are going to subtract individual observations from the mean value. So 68 minus 74, 69 minus 74, 70 minus 74, 71 minus 74, and so on. And these are the values. So 68 minus 74 is minus 6, 69 minus 74 is minus 5, and so on. So we have calculated these deviations from the mean values. Then we have to take the square of these deviations. So the square of 6 is 36, the square of 5 is 25, the square of 4 is 16, and so on. So we calculate the square of deviations. And because this is the grouped data, So we have calculated the square of deviations for these individual observation values for these individual unit steps, but we also have to multiply them with frequency because they are present as like 68 is present only one time, 80 is also present only one time, but other units, they are repeatedly present in the observation, so we have to multiply them with a frequency to take all those observations into our data. So now we have to multiply them with frequency. So this is going to be the square of deviation of the first class, which is 36, multiplied by the frequency of first class, which is 1. Then we have 2 multiplied by 25. So what is 2? 2 is the frequency of our class number 2, and the 25 is the square of deviation of this observation. So we calculate f into x minus x bar square for each of these classes. And then in the end, we take their sum, which is summation f x minus x bar square, and this is 544, right? So this is the value for summation f x minus x bar whole square. And now we are going to divide it by summation f minus 1. So we have 544 divided by 100, which is summation f, 100 minus 1, and we get the standard deviation calculation as 2.34. And we are going to write down our results as usual. So mean plus minus standard deviation shoot length is 74 plus minus 2.34. So this is the calculation for standard deviation for the grouped data. And now we are going to talk about variance, which is another important measure of variability. And variance is what? Variance is simply the square of standard deviation. And you remember the symbol for standard deviation. The symbol for standard deviation is sigma. So because variance is the square of standard deviation, so the symbol for variance is also the square of the symbol of the standard deviation. So sigma square is the population variance, and S square refers to sample variance. Like the symbol S, it refers to the sample standard deviation, so S square represents the sample variance. And standard deviation is when we take the square root of variance. So this is the relationship between these two terms, the mathematical relationship between these two terms. So because it is the square of standard deviation, so the formula for calculation of variance is simply like the formula for the standard deviation, except that if we remove the square root from the formula of standard deviation, then it becomes the formula for calculation of variance, right? So variance is summation x minus x bar whole square, which is sum of squares divided by n minus 1 for the sample variance, and for the population variance it would be summation x minus x bar square, which is sum of squares divided by the total number of observations, right? So this is going to be the same formula, except we have removed the square root from this formula. So this is the formula for calculation of variance, and this is the formula for calculation of variance when we have the grouped observations, when we have the grouped data, right? Now, where variance is used, because variance is the similar calculation as standard deviation, right? So where do we use variance? So variance is used in some statistical procedures. Like variance is used for the comparison of variability between two populations before performing a statistical analysis, and variance is also used in the comparison of mean values, like the analysis of variance. So analysis of variance basically focuses on the calculation of variance, and then these variances are compared. So variance is basically used in statistical procedures to analyze mean values and to compare the variances of two populations, or to compare the variances of two samples. So variance is used in statistical techniques. Here we have the example for calculation of variance. So you can see that we have made a similar table like the table for calculation of standard deviation, and we have taken this same example that we took in the standard deviation, or example number 1, which was about the shoot length from 10 observations, and we have the formula for the calculation of mean value. So we calculate the mean value. Mean value is calculated by dividing summation x by the n. So we have 81.4 mean value. Then we take the formula for variance, which is sum of squares divided by n minus 1. So here also we are going to take the deviation from the mean value for all the individual observations, like 78 minus 81.4, 79 minus 81.4. So this is going to be minus 3.4, minus 2.4, and so on. And then we take the square of these deviation from mean values, so which is going to be 11.56 for the first class, 5.76 for the second observation, 1.96 for the third observation, and so on. And then we take the sum of squares, and it turns out to be 56.4. And then we are going to incorporate these values into our formula, and we get the value of variance, which is 6.27, right? So if we take the square root of this 6.27, it will become the value of standard deviation. But if we stop at this point, this is actually the variance value, right? So we are going to report the variance as the mean shoot length is 81.4 millimeters and variance is 6.27 millimeters. So this is the simplest way of describing variance, right? So today we have discussed the measures of variability, and first we discussed that why measures of variability are required, because they give us an idea about the variation from the mean value. So when we calculate the measures of central tendency, when we calculate the mean value, we also need to give information about the variability, that how much the observations that vary from the mean value. And for that purpose, we use different measures of variability. The most basic one is range, which gives us an idea about the spread of observations, that how wide the distribution is from the lowest to the highest observation. And then we calculate the standard deviation, which is the standard manifestation of the variance, variability, sorry, not variance, but variability. And standard deviation is actually the calculation of deviation from the mean values, that how much the individual deviation, how much the individual observations, they deviate from the mean value. And then we have the variance, and variance is also a depiction of the variability, and it is used in certain statistical procedures, right? So these are the measures of variability.